physical interactions between different proteins?
It's more a weight of the association of the pathway and drug target with respect to drug response, actually.
Okay, so in this, where are the pathway information from?
So it's based on gene expression perturbation data. Yeah, it's like it's the pathway information is based on downstream perturbation of the certain pathway. We give a compound to pathway to perturb target the protein, and then we measure the gene expression changes.
Okay, thank you, Minyoung. Now we have Lenore Cowen from Tufts University, and she's going to talk about an update on a very nice challenge we had, which is a disease module identification challenge. Thanks, Lenore.
Okay, thank you. Good morning. Thank you for having me. Can everybody hear me? Okay. So I am reporting on the disease module identification dream challenge, which took place 2 years ago, June 24th to October 1 in 2016. And the reports were first reported at this conference in Arizona. So it makes sense to review, for those of you who weren't here then, how the challenge was structured. It was basically, I advanced and I didn't mean to. Okay. It was basically the community contributed a bunch of, I better use this pointer. The community contributed a bunch of networks. Sorry. Where's the mouse? Okay. Oh, it's backwards. Okay. So the community contributed. I can't move the mouse and speak into the mic. All right, I'm going to point with a pen. I'm low tech. All right, so the community contributed a bunch of networks. They're at the top of the slide. Okay, perfect. One was, two of them were just standard PPI interaction networks. There was a signaling network. In this case, the edges were actually directed. There was a co-expression network. There was a cancer dependency network that was based on Project Achilles. And there was a homology-based network. Of course, the challenge participants actually didn't know the type of data that was being presented or the identity of any of the gene names. So all the networks were presented to the challenge participants in anonymized form. So if the genes were IL-17, JAK, STAT, they were presented as 1, 2, 3. Okay? And there were two challenges. One in which each network was anonymized separately. So there were 6 different networks. You just had to take the structure of the network and try to produce modules. And the other was when the same persistent identifier, so you couldn't tell this was IL-17, it was only some anonymous number, but then you can follow the same gene through all 6 networks and maybe do an integrative analysis on all the types of data. The goal of the challenge was each team was expected to produce collections of modules, each containing between 3 and 100 genes. And the really novel thing about this challenge was how these modules were scored. So the challenge organizers, Daniel Marbach and his group, presented they took a huge number of GWAS data sets and this is how they broke down by types of things. This includes, for example, one on extreme height. This includes one on Crohn's disease, one on irritable bowel disease, and so on and so forth. They had all these different traits, and they took all these different GWAS studies in the literature. And what they did was basically their Pascal enrichment tool corrects for linkage disequilibrium and comes up with a score that tells you how enriched your module is for disease. So the idea is you don't know anything about disease, you're using only community detection in these, looking for sort of dense connected modules in this network. But then when they look at them, if some of your modules, the disease genes clump up in that module, that's a good module and you get a point. Okay, and there were leaderboard rounds which used a disjoint set of GWAS sets from the final test sets so that you couldn't train on the testing data but could tune parameters. And mostly I'm going to talk about sub-challenge 1. And my introduction to the project is I led the team that won sub-challenge 1 at Tufts. Team Tusk, our mascot is the elephant. But our prize is that we got to be on the paper and help analyze the post facto results. So sub-challenge 2, when they try to integrate networks, nobody did very well on. In fact, the best methods didn't do anything better than just superimposing the most informative networks or most similar networks and taking edges from all places. Okay, so I'm going to talk mostly about sub-challenge 1, as I said. This very confusing plot is a list of all the different methods that people used, and the important thing I want you to take away from this is that people used a variety of different methods. And in fact, the best performers use different methods. If you go beyond the top team to the top half dozen teams, they didn't all do the same thing. So our method is this K1 method, which is, as you can see, almost ranked top in a lot of different contexts, and we also did well at different FDRs. But there was a modularity optimization method that did very well. This is the set that includes like the Louvain algorithm and other things that are doing local optimizations of modularity. Random walk-based methods, the top one did something with walk trap. There were other methods that people used as well. And I wanted to motivate a little bit about our strategy. I started working on this problem when everybody says that the important thing in these networks is local neighborhoods, right? You want to divide your protein-protein interaction network into local neighborhoods. But there's a problem with defining local neighborhoods in these kinds of artifacts because everything is close to everyone else. It's worse than 6 degrees of separation. It's like 2 degrees or 3 degrees of separation. And in fact, if you look at the histogram of all the shortest path distances in, say, network 1, you can see that just about everything is distance 2 or 3 apart. So how can I talk about a local neighborhood when I look beyond my direct neighbors, I get basically the whole network? And if you look at the distributions on the other networks, we get something kind of similar. So our approach is in 2013, Cow et al. We introduced this DSD metric, which is a novel distance metric that is able to spread out the distances in these networks. And this was sort of our big gun. And it's a novel kernel for processing PPI networks. And our method was, on every network, we actually did the weighted version, which we call CDSD, but we included the weights of the networks. We didn't pre-process the weights. And what we did is we just applied DSD, then we used off-the-shelf methods, actually spectral clustering did best, to cluster the resulting distance matrix. And then we had to correct cluster sizes, because sometimes it would produce clusters of size over 100. We did this look for bipartite, which I'm not going to have time to talk about, and it doesn't seem to have mattered in about half the networks. But DSD spreads out the distances really nicely. You can talk about a first, second, third closest neighbor, and that makes these methods work better. Right, and once we have the distance matrix, as I said, we just used off-the-shelf methods to cluster it. All right. And so what we used the leaderboard rounds for, the only thing we used the leaderboard rounds for was training. Spectral clustering is k-means clustering in the eigenspace. All you have to tell it is how many clusters you want it to return, and we use the leaderboard rounds to set that resolution for each network. And so we got about 1,000 clusters in each network, though slightly different. Okay, I said I'm not going to talk about the used bipartite subspace. Almost all the methods that scored well recursively corrected cluster sizes. So whenever we had a cluster of size over 100, we recursively called spectral clustering again and split it so that the challenge would like it. And in some cases, we used this extra method to speed up the DSD computation, though actually it ran fairly quickly on the desktop. All right, so what do we get? How similar were the predicted modules across networks, across teams? Once we de-anonymize the data, we can ask this question and look post facto. It was interesting, across networks, across different networks, the modules were very, very different. And that might be a clue as to why Sub-Challenge 2 was hard. This wasn't just all different versions or instances of the PPI network. They might really be capturing different relationships. Within different teams, there was also not a lot of overlap. I think that comes from several reasons. One is simply the fact that the cluster contest asks for non-overlapping clusters is artificial. Makes it easier to measure performance, but if you have, everyone knows there are perfectly reasonable modules that overlap, and if you find two of them in the contest, you had to pick one arbitrarily and submit it. Also, there was no, 3 to 100 is a big range, and people did things at different resolutions. So some people picked clusters that were bigger than others. All right, so when we look at the overlap of disease genes among the different clusters, we see this picture, which is pretty much what we would expect about which diseases are more related in terms of having more genes in common. I thought, not being a biologist, I was a little surprised that type 2 diabetes and type 1 diabetes were so far apart, but everyone tells me, yeah, we knew that. Okay. And then the final thing is I was really inspired by the wisdom of crowds in the video. And so we tried as well to do some kind of crowdsourced consensus prediction. And in fact, the way we did this is also novel and interesting. We generated a consensus matrix where the similarity between two genes is defined as the percentage of top performing methods that place them in the same module. And then we ran our method, which we call the double spectral method because DSD followed by spectral clustering, is on that matrix, on that distance matrix. So, and that does better than any individual team, even our team did in the end. So, I am almost out of time. So I guess find me later if you want to know some of the interesting modules that we found. I'm particularly excited about this JAK-STAT inflammatory bowel disease module that kind of popped out. These are mostly consensus modules, but we got very similar modules in our team to these modules. We also have a very good JAK-STAT module. But JAK-STAT inhibitors are currently under study for Crohn's disease, and we find a lot of very interesting things. And we can go and look at which pathway enrichment is there for known and unknown modules. And we get some very interesting results. A lot of the pathways that are associated were known in green, but the ones in white are not known to be associated with the diseases in question. So, yeah, I want to thank the entire DREAM team, especially Sarvanaz Chubadar, Aaron Asen, Daniel Marbach, my student Jake Crawford, who is part of Team Tusk, and my collaborators in Team Tusk, Donna Slonin, Dan Hescott, Jaja Hu, Joanne Lin, and of course me. Thank you so much.